next speaker is Dr. Kratz from Australia. Okay, and she's going to speak to us about the importance of nutrition for honeybee health and colony performance during crop pollination.
Good morning, everyone. I'm Madlen. I'm a PhD student from the University of Western Australia. And today I'm going to tell you a little bit about nutrition and the effects of nutrition on honeybee health and colony performance. What my findings are showing is that there's actually more to honeybee nutrition than nutritional composition or nutritional quality. And what I mean by that is that there are other factors that we need to consider, and that, for example, includes whether the nutrition is digestible to bees in the first place. So can the bees actually make use of the nutrition or the food sources that they are feeding off? And that applies to supplementary foods, but also to natural pollens that bees forage off. So my studies are conducted in Western Australia, and some of the food sources that bees have access to, or sites that commercial beekeepers use, include canola. And beekeepers use it during our winter months, like for example now is the end of our winter, spring is starting, and beekeepers would place the colonies onto canola crops for spring build-up. So the bees build up fairly quickly on those sites. And another food source that I was looking at is clover. So clover is not a site as such, but bees still have access to clover from farmland, for example. But because beekeeping in Australia is migratory, so beekeepers on average shift their colonies to 5 to 6 different sites throughout the year, they also commonly use other sites, and these include our eucalypt forests. So one example is jarrah forest, but also Merry. And these are some of our most valuable honey crops. So beekeepers produce lots of honey. They're also a good source of pollen, so bees collect lots of pollen. And what I became interested in is in comparing the different nutrition that the bees have access to. Because ultimately, as a beekeeper, we choose the sites, but we also choose the nutrition that the bees have access to. And something else that's really interesting is that honeybees in Australia have been introduced from Europe. And so bees have evolved with some of the nutritional sources that they have access to, like for example, canola and clover, but others like our native vegetation and jarrah and Merry, bees have not evolved with. So can they actually make full use of the nutrition that they suddenly have access to? And specifically, I was interested in looking at the effects of the nutrition on the health of the honeybees, and seeing whether if beekeepers have choices between going to one side versus another, whether that makes a difference when they're then placed onto a crop. So this is an image of an avocado orchard where I placed my colonies. And I wanted to know whether there are any flow-on effects on colony health and colony performance, because bees might be very limited to the food stores that they have inside of their colonies and might rely on these if the nutrition at the orchard is minimal, or if it's mainly reliant on the crop itself. So to just quickly go through the colony setup. So I had 4 treatments with my different feeds that included jarrah, Merry, clover, and coastal. Canola, clover and canola. And for each of my different treatments, I set them up with 10 replicates, so 10 colonies for each feed, and then made up small nucleus hives of 4 frames, where I added a kilogram of bees and a sister queen to keep the genetics the same. And then also attached a pollen trap, and the reason for that was to exclude the bees from any other pollens in the environment, to make sure that the bees are exclusively feeding off the food sources that I'm providing them with. But because I did that, that also meant that I had to provide the colonies with their food sources, and the way I did that is I made up pollen paddies that were made up of pollen and the honey from their assigned food treatments. And they received 150 grams of that for anyone that's interested. So throughout the experiment was running for 12 weeks, and throughout that period, I was continuously every week providing the colonies with a paddy of their assigned food, and also taking measurements of the consumption of how much the bees were eating throughout that time. And in the first, during the first 6 weeks, during the colony build-up period, I was really mimicking the colonies having been on those sites, because they do have slightly different flowering periods, and it takes a while for the bees to build up, because the bees were made up, or the colonies were made up from scratch, so the queen had to start laying. And after the 6 weeks, the first cycle of broods of bee spawn on those beads were hatching. And after week 6, they were then moved on to the avocado orchard, and the bees arrived there just before the flowering started and at the end of the flowering period. So as I was saying, I'm interested in looking at the effects of nutrition on honeybee health, and there are various ways we can assess or measure honeybee health, and some of the measurements that I used included body composition, so looking at the body content of the bees themselves. And today I'll be specifically talking about the amino acids of bees. I also want to point out that I'm looking at emerging bees. So really looking at the effects of larval nutrition on the bees. Then I was also interested to know, are we actually seeing differences in how long bees survive? So do some bees live for longer or not so long if they are born on different food sources? And how does that relate to colony productivity? Like, how strong are our colonies? So first, let's have a look at body composition in emerging bees. And just a quick reminder, so we have our amino acids that make up our proteins, and they can be divided into two subgroups. So we have our non-essential amino acids and our essential amino acids. And today I'll be focusing on the essential amino acids, and these are the ones that bees really have to obtain from their diet, so they cannot make them themselves. For the next couple of slides or so, I'll be presenting the data that we get in the food source in terms of amino acids, and then relate that back to what we see in the bees. So what I'm interested in is seeing if the bees can actually digest the nutrients, the amino acids that they receive from the food source. So having a look at the essential amino acids as a percentage of the total protein in the food source, the first thing that I think is evident is that we see variation in the system. So there is variability between the amino acids in between our feeds. And having a bit of a closer look, you can also see that specifically Jera is lower in pretty much all of the essential amino acids. Then I wanted to know, how does that actually compare to the amino acids that the bees need and that we know of? So I started looking at, or comparing the essential amino acids that I measured to the minimum level requirements that have been reported in the literature, and that was done by Diprot. And as you can see, most of them are above the minimum requirements, so that from a nutritional or composition of the nutrition perspective, it actually looks pretty good. Once we have a closer look, you can see that there's some lower levels or closer to the minimum levels for methionine, but also in isoleucine specifically, we see that Jera is slightly lower than the minimal requirements that we know. So keeping that in mind, and I want to show you the data of the nutritional composition of the bees. And I think the first thing that I noticed that I think is very obvious is that all of the nutritional measurements of the amino acids in the bees leveled out. So no matter what they were feeding off, which food source, the amino acids in their bodies were the same. So even the low levels in isoleucine in Jera, suddenly we don't see that deficiency or low levels. So the bees are not deficient in any of the amino acids, and I think it also is a very good indication that the bees can actually utilize the nutrition and get enough of the amino acids that they need. So one way that bees can do that is by eating more of the food source, and of course that's only possible if you do not have a food source that's completely deficient in something. But if they're low levels, below the minimum requirements, bees can make up for it, and that's also what I saw in my experiments. So when I looked at the consumption of the food source, bees that were part of the jarrow-feed treatment, for example, they were eating more of the food source compared to some of the others like clover and canola. So how does that link into longevity? Do we see differences? For longevity, so at the end of the experiment, the bees were still being fed on their scientific treatments, and I was taking out brood frames to then hatch them in the incubator and make up standard feeding boxes to look at their survival or longevity. So they were kept under standard conditions, and the bees had access to water and sugar syrup. So again, I'm actually looking at the effects of larval nutrition on honeybee survival. What we can see here, and the standard way of assessing honeybee survival is looking at 50% survival or reduction in population. And what we see for Mary and Jarrah is that they essentially behave in an identical way. So after 28 days, about 50% of the bee population is still alive. When we look at bees born on canola, they seem to live a few days longer. And on clover, they kind of behave similar to canola, but then drop off. And if you look at the actual days of survival at 50%, we can see that there are significant differences. So bees born on canola actually do significantly live longer than any of the others. Now you might look at this data and think, oh, what does really a survival in a couple of days, what difference does that really make? And I completely understand why someone would ask the question, but what we actually know that in a field experiment or where bees are actually foraging, we have much more stresses on the bees. So my prediction is that in a natural environment, we would actually see much larger differences to what we see in controlled environments in the lab. So why do we see significant differences in survival if, for example, we do not see differences in body composition? And that could be an epigenetics effect, for example. So there's still a bit more to the story than what we see here. So lastly, I wanted to know how does that link in with colony productivity? Do we see differences in colony strength, especially because that's important for honey production, but also for pollination, that we do want to have really strong colonies. And to complete that, I was collecting taking photographs of frames every second week of all of my 40 colonies of each frame. And in total, I ended up with close to 2,000 images and more than 3/4 of brood cells to count. And when I talked to one of my supervisors about how we're going to do this, and he suggested to me, oh, we've got methods that we've used in the past, that's what you do. And I very quickly suggested, that's not how I'm going to do it, we need a better way of doing this. And so I got together with a friend of mine, and we developed a software called Beastly. And what Beastly does is it actually recognizes brood cells, so you can upload images onto the software, and within seconds it recognizes the cells and then gives you the actual brood count of your frames.
Right.
So that made the process just a little bit faster, as you can imagine. And if there's anyone else in the audience that think, oh my god, I wish I could use that myself, then please come and speak to me afterwards. So looking at the total brood count per colony, what we can see is, and this is over the two sites, so initially there's at week 2, that's when the colony was still growing, so the queen was still laying the first few eggs, and then we see a steep increase. But what's really, what we can really see in this graph is that clover, or bees born on clover, actually had much lower brood counts compared to any of the others. So again, what that means in terms of, for example, site selection, if a beekeeper has a choice of going to clover or going to canola or any of the others, for example, prior to pollination or honey production, we might want to have a second think about the food storage that the colonies have in the colonies and the effects on colony strength. So in summary, what nutrition is good, or how can we assess nutrition? I think we really need to think beyond nutritional composition itself and ask ourselves, how does it affect, for example, the amino acid body content? Are the bees able to digest the food source? How long do they live on the food source that they're feeding off? And how does it also affect colony productivity? Now what we can see in summary here is that canola has been, in my experiments so far, based on these measurements, outcompeting the other food sources. What I haven't mentioned today is that I'm also looking at foraging behavior, so there's still a little bit more to the story. I'm looking at foraging of pollen collectors, but also nectar collectors, and that data I'm analyzing at the moment. So we cannot only judge a food source by its nutritional composition, and with that I would like to thank all of my supervisors, industry partners and collaborators that have helped me along the way, and friends and family for the amazing support.